Okay, are there any questions? If you did not notice, I graded midterm 2A. Let me not show my screen yet. Excuse me, I'm about to sneeze here. Don't know where that came from. Well, I'm waiting for that to load. Let's go to the lesson. All right, let me show my screen now. All right, hopefully you can see my screen, and today is October 26th. Wrong month, sorry. So we should be on chapter 5, and hopefully we'll finish chapter 5 today. Please note that you'll have a quiz on Wednesday, and it will cover lab 5 and 6, and chapter 4, and the end of chapter 3. Any questions about the quiz? There is a lab today, and I'll talk more about that at 6:30. It's lab 7. All right, any question about what we're doing? If not, let's go on to the lesson. Chapter 5 is metabolism. We find where we were. So we had just talked about the different trophs, like the photoautotrophs, the photoheterotrophs, and the chemoautotrophs and the chemoheterotrophs. Any question about any of that? Let's go ahead and save the slide. You do need to know the classification system that's shown in red here. Let me blow this up so you can see it better. So you should know that we classify all organisms by their energy source. And if they get their energy needs from light, we call them phototrophs. If they get their energy needs from a chemical, like glucose, we call them a chemotroph. The phototrophs can then be split on where they get their carbon source. And if they get their carbon from CO2, we call them photoautotrophs. If they get their carbon from an organic chemical compound, such as glucose, we call them a photoheterotroph. For the chemotrophs, if they get their carbon from CO2, we call them a chemoautotroph. And if they get their carbon from an organic chemical compound, we call them a chemoheterotroph. Now we are chemoheterotrophs, and green plants are photoautotrophs. So these two should be easy for you. You have probably heard of the chemosynthetic bacteria. The chemoautotrophs can be chemosynthetic bacteria, but you do have to realize that there are some chemosynthetic bacteria which are also chemoheterotrophs. So the chemosynthetic bacteria are split between the chemoheterotrophs and the chemoautotrophs. And then the photoheterotrophs you probably never heard of. That's the green non-sulfur bacteria or the purple non-sulfur bacteria. They get their energy needs from light, but they get their carbon from an organic chemical compound such as glucose. Any question about any of that? You don't need to know that you can split the photoautotrophs into oxygenic photoautotrophs and anoxygenic photoautotrophs. And then the chemoheterotrophs can be split as well. This splitting is looking at whether they accept oxygen as their final electron acceptor, and we'll talk about that. And those are the aerobic chemoheterotrophs. And then there are some that accept their final electrons, and we'll talk about this in a little bit in the electron transport chain. The electrons don't flow to oxygen, they flow to some other molecule that's not oxygen. And those are the anaerobic respirers, meaning they perform anaerobic respiration, and they're a chemoheterotroph. You can also split, this isn't shown here, you can also split the chemoheterotrophs into those getting their, let's see, is it their energy needs? Let me think if I remember this right. They're getting their electrons from an organic chemical compound such as glucose, and that will be us. And I think I've got that shown on the next slide here. Nope, I don't. I removed it. Oh, come on. Is it here? Yeah, I removed it. If they get their electrons from an organic chemical molecule like glucose, they're the normal chemoheterotrophs. I'm trying to remember the name of those. And if the other way is those that get their chemical for the electrons from a inorganic chemical compound, and mostly that'll be the chemosynthetic bacteria, and they get their electrons from, well, it could be from an inorganic compound like ammonia or iron or a nitrogen compound or a sulfur compound. And those are called lithotrophs. Litho means rock, so troph meaning rock eater. It's close, I mean, it's an inorganic chemical. Any question about any of that? Okay, can you guys hear me?
Yes.
Okay, great. Thanks for replying. Let's see, where you talked about that. All right, let's switch gears and say that all molecules have energy associated with them. And this is especially true of the nutrient molecules. The energy is in the electrons that form the bonds, the chemical bonds of that molecule. And in cells, energy can be obtained from these bonds. And catabolic reactions break these bonds, and that can release the stored energy. In our cells and in most cells in general, energy that's needed for the cell to do work is obtained from ATP. The cell will break one of the phosphates in ATP, and that will release the energy that the cell needs for its energy needs. So we say ATP is an energy carrier of cells. It actually has two high energy bonds. A high energy means it's an unstable bond. And when the phosphate group is broken off, that releases a lot of energy, both quickly and easily. And then the cell can use some of that energy for the cell's energy needs. I'm saying some of that energy because whenever you convert energy from one form to another, some energy is always converted into heat. All right, any question about any of that? So when the cells, or our cells, whatever you want to say, produces energy, it does so by using oxidation reduction reactions. And these are two reactions that are always paired. When one molecule becomes oxidized, the other molecule becomes reduced. Oxidation is when a molecule loses its electrons. And this could be an atom that loses its electrons, meaning a molecule or an atom. Reduction is where the other molecule or atom gains an electron. So when one electron is lost from one molecule, it's gained by the other. These are always paired, meaning coupled. And we call them oxidation reduction reactions. And because that's sort of a mouthful, they came up with a shorter name, and it's called redox reactions. An example would be A loses an electron, and so A becomes oxidized. B gains the electron, and it becomes reduced. Specifically, copper +1 can get together with iron +3, and the copper can lose an electron and become copper +2. And the iron +3 gains an electron, and it can be iron +2. Any questions about that? Now, this is a little mnemonic device for helping you remember oxidation reduction reactions. You can say, Leo, the lion, goes grr. Leo, lose electrons, oxidation. Grr, gain electrons, reduction. So Leo, the lion, goes grr. Any questions about that? In biological systems, oxidation reduction reactions, of course, occur, but usually the proton, H+, is removed and follows the electrons. Remember, an H+ proton plus the electron makes the hydrogen atom. That's all the hydrogen atom is, one proton and one electron. So when a molecule becomes oxidized by losing an electron to this molecule, and this could be glucose here, an organic molecule, and then NAD plus gains that electron, the organic molecule like glucose is oxidized, it loses the electron, but it also loses a hydrogen ion, which moves to NAD plus, and then NAD plus gains the electron, so it is reduced, and it gains a hydrogen, so instead of NAD plus, it now is NADH. The point is, in biological systems, that the hydrogen ion frequently just follows the electron. Any question about any of that? All right, if there's no questions, let's move on. Biological redox reactions, meaning oxidation reduction reactions, are used in catabolism to extract energy from nutrient molecules, such as glucose. The highly reduced compounds, these are compounds with many hydrogen atoms, are degraded, catabolized to a highly oxidized compound. So glucose, C6H12O6, has a lot of hydrogens. So it is a reduced compound. And this will be degraded, catabolized into CO2 and water. And we can say glucose is oxidized to CO2 and water because it's going to lose the electrons and the hydrogen and then be converted into CO2 and water. When that happens, the chemical bonds in glucose get broken and energy will be released as that happens. And some of that energy can be trapped and then used to make ATP. These highly reduced chemical compounds like glucose contain a large amount of potential energy. And our cells use that potential energy to make ATP. If you don't believe me how much energy is in glucose, I used to have a little video on that. I could show you a beaker of glucose burning and it gives off so much heat that you have to stay, several feet away from it. But I can also mention that glucose is one of the ingredients that some of the people who make bombs use glucose for the energy of the bomb. So that tells you how much energy is in glucose. Any question about any of that? You'll note I'm not giving you a recipe for making a bomb. And to tell you the truth, I don't even know how to make a bomb, but I do know that they sometimes use glucose. Our cells use glucose to generate ATP. And we do that primarily from aerobic respiration. The energy conversion process in our cells. And I should warn you, aerobic respiration is also called cellular respiration. Those two terms mean the same thing. And aerobic respiration is actually a better term because it tells you what the cell is doing, what's going on. Cellular respiration could be aerobic respiration, meaning a cell can engage in respiration, and it could be aerobic respiration or anaerobic respiration. And so the term cellular respiration isn't the best term, at least in my opinion. Any question about any of that? So what the cell does is it combines glucose with oxygen, those are the reactants, and then it converts that into carbon dioxide and water, and some of the energy in the glucose can be released and stored to make ATP. A summary reaction for aerobic respiration is glucose, which you should know if you do not, is C6H12O6, gets together with 6 oxygen molecules, and then 38 ADP molecules, and 38 inorganic phosphate. That's what the PI stands for. So that's not phosphorus, that's phosphate. It's an abbreviation. And then that'll make 6 carbon dioxide molecules, 6 water molecules, and 38 ATP molecules. Any question about any of that? Now, this is just the summary reaction for aerobic respiration, because aerobic respiration does not occur in one step. Any question about any of that? All right. So when energy is produced by the cell to make ATP, this energy will be released by redox reactions, and then the cell uses that energy from the burning of glucose or some other food molecule to make ATP. ATP is generated by phosphorylation. Phosphorylation just means that a phosphate group is added to ADP. If you don't know what phosphate is, it's this here, PO4, and it has a 3 negative charge. ADP stands for adenosine diphosphate. So it has adenosine and then 2 phosphate groups attached to it. And then the energy that's released from the burning of glucose and the other inorganic phosphate can come together to make ATP. And ATP stands for adenosine triphosphate because there's 3 phosphates. Let me blow this up just a little bit. Get that out of here. You can notice the squiggly line here is a high energy bond. So this phosphate is attached to the second phosphate by a high energy bond. And actually the second phosphate is attached to the first phosphate with a high energy bond as well. So when the cell needs energy, it can split this bond off. And if it needed even more energy, it could also split that bond. Any question about any of that? All right. Cells have 3 mechanisms for making ATP. The first is substrate level phosphorylation, where you can transfer phosphate from one molecule to ADP. And this is usually a high energy phosphate molecule, meaning there's a high energy in this bond. And you can transfer that phosphate to ADP, and then you get this other molecule, which we're only abbreviating with 3 carbons, and then ATP. Any question about any of that? Substrate level phosphorylation actually does happen in glycolysis. And glycolysis is the first part of fermentation. So it happens in fermentation also, but in reality, it happens in glycolysis because glycolysis is a part of fermentation, as well as a part of other things like aerobic respiration. Any question about that? So ATP can be made by substrate level phosphorylation, where a phosphate is taken from one molecule and added to ADP. The second way that cells can generate ATP is by respiration. You could also call this oxidative phosphorylation, but I'm going to just use the term respiration. Respiration involves the sequential transfer of electrons from an electron donor molecule like glucose to a final electron acceptor like oxygen. And along the way, the electrons move from one molecule to another. And in each step, some of the energy in that electron is given off. And then the cell can use that energy to make ATP. Why the cell does not simply release the energy from glucose and then send it to oxygen to make ATP is that if you don't do it in a stepwise process, glucose, the burning of glucose puts off a tremendous amount of heat, and that would be so much heat that it would kill the cell. it would probably be so much heat that it would convert the cell to charcoal. So the cells release that energy slowly in a bunch of steps. And this is called an electron carrier chain because the electrons flow from one molecule and then to the next, and then to the next, and then to the next. So electron transport chain. It's not showing, but these electrons start from glucose. And we'll show it in just a little bit. Those electrons then move to NADH, and then they're given to this molecule, which you don't need to know, and then to this molecule, et cetera, all the way down the chain. And in aerobic respiration, the electrons flow to oxygen. All of these are sequential redox reactions, meaning one molecule is oxidized, losing the electrons, and the other molecule is reduced, gaining the electrons. The third way that cells can generate ATP is by photophosphorylation. And only photosynthetic organisms can perform photophosphorylation. You can call it photosynthesis if you wish. It's only in photosynthetic organisms like green plants, but also green bacteria. What happens is light releases the electrons from chlorophyll, and we'll talk a little bit more about that later, but light is energy, and when the energy from light is absorbed by the chlorophyll, it can release the electrons. The light energy from the electrons being released from the chlorophyll will then be converted to chemical energy via an electron transport chain. We'll talk more about that at the end of this lesson. And along the way, ATP and NADPH are generated. In photosynthetic organisms, ATP and NADPH are often used to make sugar. Any question about any of that? And this is a picture showing you some photosynthetic organisms. That's why they're so muddy. Only kidding. Although there are photosynthetic organisms in the background. Any question about any of that? The point is cells can generate ATP in three ways: substrate level phosphorylation, by respiration, and by photophosphorylation. The energy that's released from molecules is released in controlled ways and stored by a series of reactions. It's not a single energy burst, as I stated, because if you were to burn, like I said, a beaker of glucose, right, I guess you can't see me, let me bring that up a little bit. Right here, I couldn't be here if the glucose were that close to me. I'd have to be like 6 feet away because it releases so much energy. And how the cells do that is the electrons from the donor molecule are passed from one compound to another through a series of redox reactions, releasing that energy slowly. Many cells can perform carbohydrate catabolism, and they use that as their primary source for obtaining cellular energy. Most organisms can do this. In fact, even green plants can obtain energy from carbohydrate catabolism. You probably don't know this, but like in Point Barrow, Alaska, in the wintertime, when the sun sets for 3 months out of the year. So how do plants survive 3 months without any light? Anyone have a guess?
Do they like store it away maybe for later use?
Yeah, what they do is they make glucose and other molecules, and then they store that. And then in the wintertime, when they have no light, they use carbohydrate catabolism to obtain their energy needs. And actually, even green plants in your house, assuming you don't have a plant light, they will perform carbohydrate catabolism at night to obtain their energy needs. So in the daytime, they'll use photosynthesis to get their energy needs. But at nighttime, green plants, as well as any green bacteria, will use carbohydrate catabolism to get their energy needs. Of course, for us, we almost totally use carbohydrate catabolism to get our energy needs. And if we don't use carbohydrate catabolism, we get our energy needs from digesting protein or lipids. And they're pretty similar to carbohydrates. In fact, for most of us couch potatoes like me, when I eat a high protein diet or a high lipid diet, I will convert the protein and the lipids to carbohydrates, and then they'll be converted to glucose. And then glucose will be used in the normal way to obtain my energy needs. That is an exception for a marathon runner, that they can digest lipids directly. However, even a marathon runner will not digest lipids for the first 45 minutes of their run. In the first 45 minutes of their run, they will be using carbohydrate catabolism to get their energy needs. And only after 45 minutes in their run will they switch to digesting lipids and then using that to get their energy needs. Any question about that? And that's assuming, of course, that a marathon runner is not eating while they're running. But you can use lipids and proteins as well as carbohydrates to obtain your energy needs. Glucose is the most common energy source for organisms that perform carbohydrate catabolism. Organisms that get their energy needs from glucose have two general processes for getting their energy from glucose. They can perform respiration, or they can perform fermentation. Now with respiration, there's two types of respiration. There's aerobic respiration, and there is anaerobic respiration. We'll mainly talk about aerobic respiration, but once we're done talking about aerobic respiration, we'll talk a little bit about anaerobic respiration. So let's watch a video on aerobic respiration. Assuming that it loads.
Hey, I'm Alex Honnold, and to support the work of my
all cells need energy to stay.
Let me turn up the volume, because this is really quiet. Oops, that's the wrong one. Come on.
Stay alive. Usually, all cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
Okay, any questions about that video?
No, I think we're all good.
Okay, good, thanks. All right, as I stated, cells can obtain their energy needs if they're using carbohydrate metabolism from either respiration or fermentation. We're going to start by talking about aerobic respiration, and then we will talk about anaerobic respiration. So our cells can obtain it from both aerobic respiration and fermentation, although the only cells of ours that can perform fermentation are your muscle cells. This slide shows you an overview of aerobic respiration and fermentation. Fermentation starts with glycolysis, and you take the glucose, which you can barely see right there, and it'll be converted to pyruvic acid in glycolysis, and then the fermentation steps occur, so that glycolysis and then the fermentation steps, it's usually 1 or 2 steps occur. And then the pyruvate is converted to a fermentation product like ethanol or lactate, and ATP is generated. The ATP is only generated in glycolysis. Aerobic respiration has 3 steps. It too starts with glycolysis, and that makes the pyruvate, which then goes to the Krebs cycle. And then the Krebs cycle burns the glucose and sends the electrons via an electron carrier like NADH to the electron transport chain, or ETC. And then the electrons flow from essentially glucose to oxygen. So oxygen is the final electron acceptor, why it's called aerobic respiration. And the glucose is converted into CO2 and water. This process yields a very high amount of ATP. The fermentation is very low on yielding ATP. Any question about any of that? Another slide of aerobic respiration in a eukaryotic cell. So the pink would be, or light purple would be the cell. And this is a eukaryote because we have a mitochondria. Glycolysis always happens in the cytoplasm, where glucose is converted to pyruvate. And then the pyruvate moves in our cells into the mitochondria and enters the Krebs cycle or the citric acid cycle. You do need to know the Krebs cycle has a couple of names. And then the electrons go from the citric acid cycle or the Krebs cycle to the electron transport chain, which also has a large number of names, including oxidative phosphorylation. I will not call it oxidative phosphorylation. I will call it electron transport chain because that's what your textbook uses and it's what I use too. Anyways, along the way, ATP gets made, glucose is converted into CO2 and water. The point here is glycolysis happens in the cytoplasm. In a eukaryote, the Krebs cycle and the electron transport chain happen in the mitochondria. In a prokaryote, the electron transport chain happens at the cell membrane, and we'll talk about that. All right, so glycolysis has another name, but you don't need to know it. I never use it, and I think it's mentioned one place only in the book. Glycolysis is the oxidation of glucose to pyruvic acid. You can find glycolysis in most cells. Glycolysis can occur in the presence or in the absence of oxygen. It does produce ATP and NADH, but not very much. All right, any questions about glycolysis in general? This slide is showing you the different steps of glycolysis, a 9 or 10 step reaction, however you count it. Where glucose has 6 carbon atoms, is converted into pyruvate, which has 3 carbon atoms. And there's 2 of them, 2 pyruvates that get made from 1 glucose. And that's why we've got 6 carbons when you multiply by 2 pyruvates. Note that right here, this molecule is converted to that molecule and then it goes down this reaction. ATP does get made right here and here, but it does take ATP to get there, meaning you take in ATP here and here. So the net gain of ATP from glycolysis is 2. There are 2 ATP generated from glycolysis for each molecule of glucose. When I was in college, I had to know the names of all the intermediates, as well as the enzymes that made these intermediates. And I think it's fair that since I was required to know those, that you should be required to know those too. What do you guys think? So you think it's fair? Okay, well then I will hold you if you're knowing that. So this is a summary reaction for glycolysis. And that is you have glucose, 2 ATP, 4 ADP, 4 phosphate, 2 NAD plus, generate 2 pyruvic acid, 4 ATP, 2 NADH, 2H plus, and 2 water. You don't really need to know this, but you should know that we start with glucose and you end up with ATP, but you get a net gain of 2 ATP and 2 pyruvates come from the glucose and 2 NADH. I should mention that pyruvate and pyruvic acid are very similar molecules. And our cells will treat them the same. But if you're a chemist, they are different. The pyruvic acid has the hydrogen attached. The pyruvate does not. And you'll find that in one place I call it pyruvate because that's what the slide has. Generally, I call it pyruvic acid, which some of the slides have. Any question about any of that? All right. So respiration is the oxidation of molecules to release the energy and the electrons. In aerobic respiration, the final electron acceptor, what accepts that electron coming from glucose, is oxygen. In anaerobic respiration, the final electron acceptor is not oxygen. It's a molecule other than oxygen. And it depends on what form of respiration it is to determine what that molecule is. But it could be a sulfate, or it could be a nitrate, or a carbonate, or an iron. It could be a lot of things because there's many different forms of anaerobic respiration. But there's only one form of aerobic respiration. In the way respiration generates ATP by oxidative phosphorylation. So this is just a little summary of what respiration is. Let's talk about aerobic respiration in greater detail. The pyruvic acid comes from glycolysis, but pyruvic acid cannot directly enter the Krebs cycle. It's therefore oxidized and decarboxylated, and pyruvic acid is converted into acetyl coenzyme A. The pyruvic acid is a 3-carbon molecule. Acetyl coenzyme A is a 2-carbon molecule. So one of the carbons of glucose comes off from pyruvic acid at this step, releasing CO2. We generate NADH also. Now remember, glycolysis creates 2 pyruvic acids. So the preparatory step will run twice for each molecule of glucose. Any question about any of that? The acetyl coenzyme A can now enter the Krebs cycle. You should know that the Krebs cycle is also called the tricarboxylic acid cycle or the citric acid cycle. And that's usually abbreviated TCA. It's called the Krebs cycle because Dr. Krebs discovered it. Your textbook calls it the Krebs cycle, and that's the way I learned it, so I call it that as well. In the Krebs cycle, and this shows you the preparatory step and then the Krebs cycle here. Let me blow this up. In the Krebs cycle, the acetyl coenzyme A joins with oxaloacetic acid to form citric acid, which you don't need to know that name there. And then that molecule changes to that, to that, to that, to that, to that, to that, ending up on oxaloacetic acid, meaning we're ending up with the molecule we started with. Come on, mouse, come back. And that's why it's called the cycle, because this will just keep going around and around and around as long as acetyl coenzyme A comes in. Along the way, we generate NADH in the Krebs cycle, one here, and one there, and in the preparatory step, one there. So that 4 get made, 4 NADH per molecule of pyruvic acid. And 1 ATP gets generated right here for each pyruvic acid. And then 3 CO2 come off, and that's all of the carbons in pyruvic acid. One comes off in the preparatory step, one comes off right here, and the other comes off right here. Now remember, glycolysis generates 2 pyruvic acids. So the preparatory step and the Krebs cycle will run twice for each molecule of glucose. Any question about any of that? All right, so you need to know what's in red here. You should know you start with acetyl coenzyme A. And then you end up really with CO2. And of course, NADH, 4 NADH and 1 FADH2. I haven't talked about that. That comes off right here. The NADH right here, right here, right here, and here. All right, any questions about this? So how many molecules of ATP are generated by the Krebs cycle for each molecule of glucose? Nobody's gonna guess. All right. This runs twice, so you generate 2 ATP. You guys really do need to participate more. All right, I'm showing you the overview of respiration again. We're going to go on. The NADH and the FADH2 carry the electrons from glucose and then take them to the electron transport chain. In the electron transport chain, the production of ATP kicks into high gear. Let's watch a little video on that. The ETC does have a couple of names. I generally call it the electron transport chain, or you call it electron transfer chain. And then there's electron transport phosphorylation. Students don't tend to get confused on those. I do not call it oxidative phosphorylation. That term is very different, but it can also be called that.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation.
All right, any questions?
This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Oh, come on. Can't close that down. That's on the way. All right. Let's come back. So in eukaryotes, the electron transport chain occurs along the inner mitochondrial membrane. So this right here is where electron transport chain occurs, and it's the inner mitochondrial membrane. It's called the electron transport chain because the electrons transport from NADH to this molecule, to that molecule, to that molecule, to that molecule, to that molecule, and then finally to oxygen. So it's a chain of proteins in a membrane, and it's called the electron transport chain. In prokaryotes, they don't have a mitochondria, but the electron transport chain is a chain in a membrane. And what they do is they do it at the cell membrane. So these proteins are in the cell membrane. And the NADH brings the electron to the cell membrane. And then the electron's transferred, ending up at oxygen being the final electron acceptor in aerobic respiration. Along the way, let's blow this up a little bit. Along the way, as the electrons move through some of the proteins, including this one and this one and that one, the electron gives the protein enough energy to pump hydrogen ion from this side of the membrane to that side of the membrane. That will mean there's less positive charge over here and then more positive charge over there. There will also be more hydrogen over here than here, and that creates a twofold gradient, meaning there's more positive charges here than here and more hydrogen here than here. And that's potential energy difference. These H pluses are going to want to move by the laws of diffusion from this side of the membrane to that side of the membrane. Of course, they can't do that. They can't move through the lipid bilayer except at this channel protein. And as the hydrogen ions move through this channel protein, it gives this protein the energy it needs to make ADP into ATP. So this channel protein is an ATP synthase molecule. And that is how the cell makes ATP, or at least the majority of the ATP, because the majority of the ATP is made in the electron transport chain. Now, what happens to the electrons in the hydrogen ion? They move from glucose, which isn't shown here, to NADH or FADH2, And then the electrons, as well as the hydrogens, come off here. The electrons move from one protein to another, the electron transport chain, and then they flow to oxygen. So oxygen plus 2 electrons plus 2 hydrogen ions make water. And that is why you need to breathe. All the oxygen you breathe will be converted by aerobic respiration in your mitochondria into water. The body can use some of that water, and a lot of it does get breathed out. None of the oxygen is converted into CO2. Okay, all the CO2 that we breathe out happens from the Krebs cycle in the preparatory step in the burning of glucose. So glucose is converted into CO2. The oxygen we breathe in is converted into water because of the electron transport chain. Any question about any of that? All right, as I stated in aerobic respiration in the electron transport chain, the production of ATP kicks into high gear. In eukaryotes, the electron transport chain makes 32 to 34 molecules of ATP for each molecule of glucose. Prokaryotes are a little more efficient and they generate 34 molecules of ATP for each molecule of glucose. You don't really need to know this, but the cells in your body which are more efficient and generate 34 molecules of ATP are your heart, your liver, and your kidney. All the other cells in your body, including your brain, your skin, your nose, they only generate 32 molecules of ATP for each molecule of glucose. Why do you think your heart, your liver, and the kidney are more efficient and generate 34 compared to the cells of your nose or your skin? If nobody answers, I'm going to say your brain cells also only generate 32 molecules of ATP. Why does your brain cell use less than your heart or make less than your heart? Well, as is obvious, your brain cell is not working right now, and your heart cell hopefully is, otherwise you'd be dead. So your heart, your kidneys, and your liver are always working. So those cells are more efficient at generating ATP. And the prokaryotes are more efficient. In aerobic respiration, the final electron acceptor is oxygen. And so we call that the final electron acceptor. It accepts the electron that flows from glucose. All right, any question about any of that? This shows you what I was talking about previously. The electron transport chain, where NADH brings the electrons to one molecule. You don't need to know these molecules, but you do need to know that NADH and FADH2 bring the electrons to the electron transport chain, and then the electrons flow to oxygen. This is showing you, let me blow this one up. In a eukaryote, this is the inner mitochondria membrane where the electron transport chain occurs. So it's occurring across this membrane. The Krebs cycle is happening inside the inner mitochondrial membrane. In prokaryotes, they don't have a mitochondria. And so this electron transport chain is in the cell membrane. Now for eukaryotes, they pump the hydrogen ion from the inner mitochondrial membrane into the space between the inner membrane and the outer membrane. Meaning in eukaryotes, we have this all compartmentalized, and it's there to protect the cell. In prokaryotes, the hydrogen ion is actually pumped outside of the cell membrane and into the cell wall. So this region here will be the cell wall. And then these hydrogen ions will come through this channel protein to make ATP. Any question about any of that? We already talked about that. Just making sure. This exact step of the hydrogen ions flowing through ATP synthase to make ATP is called chemiosmosis. I don't really use that term, but that is something that another instructor may use, and I think your textbook talks about it. So I'll mention it. And then the electron transport chain can be called oxidative phosphorylation. As I stated, I don't call it that, and your textbook doesn't either. There are a lot of terms that mean the same thing. All right, let's count up the ATP we get made from aerobic respiration. We get 2 from glycolysis. We get 2 from the preparatory step in the Krebs cycle. And then we get 34, 32 to 34. 34 in prokaryotes, 32 to 34 in eukaryotes. So the total amount of ATP is 36 to 38 ATP per molecule of glucose in eukaryotes. And in prokaryotes, it's 38 ATP per molecule of glucose. Here's the summary equation for aerobic respiration, which I've already given to you. C6H12O6 plus 6 oxygen molecules, 38 ADP, 38 phosphate, generate 6 CO2, 6 water, and 38 ATP. Any question about any of that? All right, we're going to stop right here, and I'll continue with this in our next lesson. And I'll see you at 6:30 for the lab.